Hi everyone, I'm Xiaoyue Xu, a PhD student from CSBL. Today my presentation title for the journal club is A Swapped Genetic Code Prevents Viral Infections and Gene Transfer, which published on Nature recently. Here are the highlights of the journal. In this study, researchers created a new type of biocontained E. coli strain based on Sin61 delta 3 cells, which genetically re-engineered alternative genetic code to prevent viral infections and horizontal gene transfer. And they developed cells bearing an amino acid swapped genetic code that reassigns 2 of the 6 serine codons to leucine during translation. This amino acid swapped genetic code renders cells resistant to viral infections by mistranslating viral proteomes and prevents the escape of synthetic genetic information by engineered reliance on serine codons to produce leucine-requiring proteins. The conservation of the genetic code permits organisms to share beneficial traits through horizontal gene transfer and enables the accurate expression of heterologous genes in non-native organisms. However, the common genetic code also allows viruses to hijack host translation machinery and compromise cell viability. Additionally, genetically modified organisms can release functional DNA into the environment. Virus resistance and biosafety are among today's major unsolved problems in biotechnology. And no general strategy exists to create genetically isolated or virus-resistant organisms. Furthermore, biotechnology has been limited by the 20 amino acids of the canonical genetic code, which use over 64 possible triplet codons, limiting efforts to expand the chemical properties of proteins by means of non-standard amino acids. Changing the genetic code could solve these challenges and reveal new principles that explain how genetic information is conserved, encoded, and exchanged. Scientists propose that genomically recorded organisms would be genetically isolated from natural organisms and viruses, as horizontally transferred genes would be mistranslated, producing non-functional proteins. Furthermore, genetically recorded organisms could provide dedicated codons to improve the purity and yield of non-standard amino acid containing proteins, enabling robust and sustained incorporation of more than 20 amino acids as part of the genetic code. First, I will briefly introduce about the background. Naturally, there are 64 genetic codes as shown in the slide. Genetic code refers to a nearly universal assignment of triplet codons of nucleotides to amino acids, linking hereditary entities to the functional blocks of life. In practice, this codon to amino acid assignment is realized through the agencies of the code adapter molecules of transfer RNAs, tRNAs, with a codon's complementary replica anticodon and the corresponding amino acid attached to the 3' end. In this study, 2 serine codons TCA and TCG together TCR and the TAG stop codon were placed with synonymous alternatives to prevent viral infections and HGT. In 2013, LaJoie and their colleagues constructed a GRO in which all instances of UAG codon have been removed, permitting the deletion of release factor 1 to permit efficient and site-specific incorporation of non-standard amino acids into proteins. That is, UAG has been transformed from a nonsense codon to a sense codon, provided appropriate translation machinery is present. They selected UAG as their first target for genome-wide codon reassignment because UAG is the rarest codon in E. coli MG1655. Previous studies demonstrated the feasibility of amino acid incorporation at UAG, and a rich collection of translation machinery capable of incorporating NSAAs has been developed for UAG. As a result, modified E. coli provides substantial but not complete resistance to bacteriophages. After that, Sin61-3 was created with a synthetic recorded genome in which all annotated instances of two serine codons TCG-TCA and the TAG stop codon were replaced with synonymous alternatives, and the corresponding serine tRNA genes and RF1 have been deleted. Here is the way to create Sin61-3 according to the Robertson article published on Science in 2021. First, they replaced the three codons TCG, TCA, TAG. Then they found the growth rate of the Sin61 was slower than before it derived. So they applied 2 rounds of parallel mutagenesis to increase the growth rate and selected the fast growing strain, and equal Sin61F2 in this stage. And after that, they removed the SIR-T, SIR-U, and the PFRA to create Sin61-3. And again, the growth rate decreased compared with the Sin61F2, so they applied 3 more rounds of parallel mutagenesis and selected the fast-growing strain, and equaled Sin61-3F5. Although the compressed genetic code of Sin61-3 provided resistance to 5 viruses, it could not prevent the escape of its engineered genetic information. Since reduction to 61 codons demanded that a whopping 18,214 codons be changed, necessitating a genome synthesis approach. Freden and colleagues built their synthetic E. coli genome by using large-scale DNA assembly and genome integration methods that they had developed previously to prove the limits of codon changes in E. coli. In their approach, DNA is computationally designed, chemically synthesized, and assembled in 100 kilobase fragments in vectors in S. cerevisiae, and these vectors are then taken up by E. coli and integrated into genome in the direct place of the equivalent natural region. Iterating this process 5 times resulted in 500 kilobase sections of DNA being replaced by synthetic versions. 8 strains of E. coli were produced in this way, each harboring synthetic DNA sections that covered a different region of the genome. These sections were then combined using conjugation methods to make the complete synthetic genome. And authors investigated whether tRNAs of mobile genetic elements can substitute cellular tRNAs and support the viral infection in cells with a compressed genetic code. They screened thousands of mobile tRNA for tRNAs that can simultaneously translate TCA, and TCG codons by computationally identifying tRNA genes in mobile genetic elements, and then synthesizing 1,192 candidate tRNAs as an oligonucleotide library and cloning these variants into a plasmid vector carrying a MPT2 marker conferring kanamycin resistance, containing TCR codons at positions 40 and 104 and TCG codons at positions 68 and 251. Following the transformation of this library into Sin61-3, in which the deletion of SIR-U and SIR-T makes TCG and the TCA codons unreadable, only variants carrying functional TCR suppressor tRNAs survive kanamycin selection. Finally, high-throughput PCR amplification and the sequencing of the tRNA inserts from kanamycin-selected clones and identified 62 tRNA sequences capable of promoting MPT2 expression. These tRNAs represent 89% of all predicted TCR codon recognizing tRNAs in their library and share 33.7 to 61.1% similarity to the endogenous CERU tRNA of E. coli based on the multiple sequence alignment mobile TCR codon translating tRNAs as shown in Figure B. Consistent with the anticodon composition of mobile serine tRNAs, most tRNA hits contained a UGA anticodon as shown in gray shading and carried the identity elements necessary for recognition by the host serine tRNA ligase as shown in blue. The researchers confirmed the identity of the TCR codon recognizing tRNAs by co-expressing a selected tRNA hit with a specific construct and found that mobile tRNA-derived tRNAs can promote viral replication. They discovered that diverse TCR codon translating tRNAs on horizontally transferred genetic elements indicate that mobile tRNA genes are widespread and can substitute cellular tRNAs to promote viral replication and HGT. Their study found that tRNA genes that reside in viral genomes can replace cellular tRNAs and promote phage infection, which was demonstrated by the infection of Sin61 delta 3 harboring a bacteriophage of derived cell tRNA UGA gene with T6 resulting in rapid lysis. Next, to investigate the existence of lytic viruses in Sin60 delta 3, the authors conducted an experiment to isolate viruses from environmental and wastewater samples using Sin61 delta 3. They first mixed the samples with Sin61 delta 3 and grew them until they reached the stationary phase. Then removed the bacterial cells and tested for the presence of lytic phages by creating virus-infected areas on soft agar overlays. Through this process, they were able to identify 12 new phage strains belonging to the Cordovirus order and Myoviridae family, all of which had tRNA operands encoding 10 to 27 tRNA genes in each genome. Additionally, one of the strains, REP1, also had a predicted homing endonuclease. Homing endonuclease encoded in tRNA operands have been shown to be responsible for the horizontal transfer of tRNA gene clusters. The authors also found that the viral titers after replication on unrecorded cells differed by more than 2 orders of magnitude among the different strains, with REP12 being one of the most virulent strains, taking 60 minutes to complete a replication cycle at 37 degrees in Sin61 delta 3. The study found that there are bacteriophages in environmental samples that are able to overcome viral resistance because by the recording of sense codons. However, even though the viral genomes also contain the tRNAs that can translate the recorded codons, the presence of these tRNAs did not directly rescue viral replication in the recorded strain. And finally, the study also verified the function of phage tRNAs in decoding TCR codons. The researchers cloned the REP12 viral tRNA operand containing the predicted tRNA cell UGA and its promoter into a plasmid vector. Co-expression of the tRNA operand with the 2 different constructs containing either a TCA or TCG codon at position 16 resulted in high expression of elastin SFGFP with his tag. Triptych digestion and by reverse phase LC-MS analysis confirmed that both TCA and TCG codons were decoded as serine by this elastin 16 TCR SFGF with his tag samples. Similarly, co-expression of the same construct with the viral tRNA cell UGA also led to the decoding of TCA codons as serine. As confirmed by LC-MS analysis, together these results show that lytic phages of Sin61-3 overcome genetic code-based viral resistance by rapidly complementing the cellular tRNA pool with the virus-encoded tRNAs. The researchers hypothesized that by creating an artificial genetic code in which TCR codons encode a different amino acid from their natural serine identity, it would create a genetic barrier that would protect cells from HGT and infection by tRNA encoding viruses. They believed that the level of genetic isolation provided by amino acid swapped genetic codes would depend on the differences in hydrophobicity and polarity between the exchanged amino acids. To test this, they attempted to reassign TCR serine codons to leucine, which is the amino acid that is most different from the natural serine meaning of the TCR codon. To establish a serine to leucine swapped genetic code, researchers utilized Sin61-3, which genome-wide lacks annotated instances of TCR codons and their corresponding tRNA genes, and sought to identify tRNAs capable of efficiently translating TCR codons as leucine. The creation of a genomically recorded organism, EC61-3SL, from E. coli, in which both TCA and TCG codons are translated as leucine instead of serine. The introduction of bacteriophage-derived LU-TCR UGA and LU-tRNA CGA to Sin61-3 reassigns TCA and TCG codons to leucine, and the reassignment of the TAG stop codon to encode BIPA in an essential gene of the host ensures the biocontainment of Sin61-3SL. Here, researchers screened tRNA library to evolve TCR suppressors from endogenous E. coli LU tRNA carrying a TCA and TCG decoding anticodon. The researchers created a mutagenized library of the LU tRNA gene that had a fully randomized anticodon loop, which consisted of 65,536 different variants. This library was co-expressed with a kanamycin resistance conferring gene called APH3LA, which had all 29 instances of leucine codons replaced with TCR serine codons. The system was designed such that only the anticodon swapped LU variants capable of simultaneously translating all 29 TCR codons as leucine would confer resistance to kanamycin. The researchers expected that a high kanamycin selection pressure combined with the presence of the 29 TCR codons in APH3LA would select tRNA variants that could efficiently translate the TCR codons at wild-type levels. And Figure B shows the predicted mechanism of phage resistance in Sin61-3SL. The reassignment of the sense codons TCA and TCG to leucine in Sin61-3SL provides multivirus resistance by mistranslating the viral proteome. Next, according to the previous infection assay, phage tRNA YGA expressing cells were infected with a mixture of 12 distinct lytic phages of Sin61-3 at a multiplicity of infection of 0.1. YGA here represents UGA and CGA. The analysis of phage titer in cultured supernatants after 24 hours showed a marked drop compared to the input phage inoculum, suggesting that anticodon-swapped viral leucine tRNAs block phage replication. Untargeted deep proteome analysis of uninfected cells by tandem mass spectrometry validated the translation of TCR codons as leucine in EC-SYN61 delta 3SL. Time course untargeted proteome analysis after bacterial phage infection revealed extensive mistranslation at TCR codons in newly synthesized phage proteins, indicating that an amino acid swapped genetic code broadly obstructs viral protein synthesis. And finally, the researchers developed a set of plasmid vectors that depend on TCR codons to express leucine-containing proteins and can only function in cells that efficiently translate TCR codons as leucine. These plasmids, called the PLS plasmids, offer four widely used orthogonal antibiotic resistance markers for stable maintenance in EC-SYN61 delta 3SL cells using their PLS plasmids. The authors achieved serine to leucine codon reassignment simultaneously at about 14,000 codon positions in a single cell, demonstrating the exceptional codon reassignment efficiency of their viral tRNA-based system. The researchers tested their PLS plasmids to ensure they do not function in wild-type E. coli K12 MG1655 cells by electroporating plasmid DNA from 6 variants into these cells and found no escapees carrying PLS plasmids. The expression cassette for phage tRNA LU YGA was encoded with the plasmid backbone, which also prevented the escape of PLS plasmids, indicating that anticodon-swapped viral tRNAs are toxic to wild-type cells. The authors suggested that any leucine-requiring protein or even entire chromosomes can be addicted to EC-SYN61 delta 3SL by recording target genes to encode one or more leucine positions at TCR codons. In this study, researchers created a new type of biocontained E. coli strain, EC-SYN61 delta 3SL, carrying an amino acid swapped gene code to achieve unprecedented gene transfer resistance by the reassignment of TCR codons to leucine. The potential limitations of the work include the sampled viral diversity and the inability of viral resistance assays to sample multi-step long-term evolutionary process of viruses. And the authors suggest that the future work should explore whether such viruses can evade the amino acid swapped genetic code of EC-SYN61 delta 3SL. This is all of my presentation. Thank you for listening.